Thank you for joining us today for the HPL seminar. Today we have the pleasure of hearing from Dr. Douglas Garrett, who is a PI at the Lifespan Neural Dynamics Group at the Max Planck Institute for Human Development in Berlin, Germany. Dr. Garrett's educational background includes an undergraduate degree from the Department of Psychology at the University of Victoria in 2003, where he did an honors with distinction, followed by a master's degree also in psychology from the University of Toronto from 2005 to 2007. And following the master's degree, Dr. Garrett also went on to do a PhD in the Department of Psychology at the University of Toronto in cognitive neuroscience under the supervision of Dr. Cheryl Grady. His dissertation was titled Establishing Relations Between Bold Variability, Age, and Cognitive Performance. When I asked Dr. Garrett about some fun facts about him, he mentioned a few that were really interesting. And so to start off, he was born in Grand Prairie and grew up mostly in Lacombe, Alberta. But one that caught my attention the most was that his Berlin-born twin 8-year-old boys now try to dominate the Calgary soccer scene by pretending that they can only speak German during their games. Today, Dr. Garrett will be presenting on moment-to-moment brain signal variability as a principal signature of aging and cognitive performance.
Yeah, I can hear myself, so hope you can hear me too. Thank you very much for the chance to be here today. It's sort of like a homecoming in many ways this last few months because growing up playing all my sports around central Alberta, the biggest task in our lives was beating the guys from Calgary, you know. So pleasure to be here in the heart of athletic country. Tell you a little bit about what we've been doing the last several years. So for the last 15 years, I would say the problem that has been most important to me in science is this weird thing about the brain and the whole body, it turns out. The brain especially, that the natural state of it is instability, not the opposite. So if you measure that, here's a cartoon of someone catching a ball, right? At every level of this chain, from the sensory channel all the way through to the spindle level to catch that ball, there's massive amounts of stochasticity. But somehow that person catches that ball. Right? So why is that? How can we live this life we live in a functional way in the presence of all of these dynamics? Because I've been most focused on the brain over these years, if you just read out brain signals, it doesn't matter how you measure them. The variability is extraordinary. So in a mouse, you can present them with the identical stimulus one after another, and you'll get very different response profiles. If you average those response profiles, you get something that looks like the waning of those dynamics. But those trial-wise, one-shot dynamics never go away. Right? You're forced to grapple with how they can be there. In humans, it's the same. So no matter what kind of brain signal you measure, here it's electric fizz, it just never stops, right? So in the brain, there's no zero. There's absolutely no concept of a zero because the firing never ends. So you just have this flow that goes on, and you can ignore it, but nevertheless, it is there. Turns out not a very new problem. Already in the 60s came a very provocative review, which meant there were decades of work before that, here by Lawrence Peniel arguing in no uncertain terms that this variability is really not to be considered noise. We should rather think of it like the tone of the entire enterprise. Everything we do, everything we can process, everything we can execute is built on the backbone of dynamics, right? The tone of this variability is the substrate that we should consider. Okay. Didn't go very far, turns out, in neuroscience. There's a tendency to want to average away all of those dynamics because you're trying to get to the real signal. But this case was made already way back then. So why isn't there? Well, there's lots of work on the benefits of those dynamics, that you really should have it in the system. For example, some nice modeling work where networks that are formed in the presence of noise are more robust to disruption. So it takes the edge off the importance of any node in a network, allowing it to be robust, that if you take a node out, the thing can still survive. Noise is one of the mechanisms that allows that to happen in that model. It also is perceived to allow you to explore more neural states, potentially enhancing learning and environmental adaptation. The dynamics themselves are the thing that allow you to move internally. Another idea. Okay. So there's been some sort of thinking around this, like what is it for as a general property or mode of the brain? Well, we don't know what we're going to get every minute of the day, do we? Right? What is the basic mode of function we should have if we didn't state lock our brains? That is, if we overfit the world, we say, okay, this minute, no matter what, I think X is going to happen. If you lock down to that mode, you're not very responsive to the reality of what actually happens. So take Nadal, what does he do when he's waiting for a serve? Does he sit like this and then move and act? It's incredibly difficult physically to do that. So he gears himself up in this hippie hop way. And it turns out each player on the ATP tour does something a little bit differently. There's nothing stereotyped about this action. So it's a mode of preparation that he uses. And maybe the brain does that too in a general way that's unique to you. So one way to think about it is maybe this dynamic range or degrees of freedom is a property that allows system flexibility to exist. If we lock everything down, we lose that ability. So the group in Berlin basically focuses on this problem in a bunch of different ways. From what happens across the lifespan. Cognitive performance is a core part of what we do. So really trying to understand when people do well, when they remember things well, process the world well. How does variability lead into that? Connectivity, stimulation, computational modeling, we cover a lot of bases. I'm just gonna talk about a few of these today. Good time. So what do we mean actually? We read out these brain signals. There's a million ways you can think about variance in any scientific domain. In neuroscience, there's a few kind of common ways that it's considered. It's sort of ramp up, let's say, in details. So one is you can take the distributional width, right? Just a simple variance of that signal. This is a raw signal. Another way is to compute the spectral properties of that. So instead of the overall variance, it's about, if you do a Fourier transform, you have a lossless transform from the raw time domain into oscillations. This is another way you can conceive of the world of variance. Or you could take a general mindset of entropy. And entropy is basically about counting patterns, right? How repetitive a signal is. If you have a perfect oscillation, it will have zero entropy, basically, because you have the repetition of that thing over and over. But most signals aren't really like pure waves, right? They're made up of complex dynamics. So entropy will get you around this kind of problem. So in human aging, this has become a core interest of mine over the years, based on, at least partially because of its history. So this idea of noise in the brain is something that was already cooking in the 50s. Crossman and colleagues, for example, that the reason your brain doesn't work as well when you get older, you don't remember as well, for example, is because there's some sort of mystic noise in that brain. However, there was no studies of the brain when those ideas came to be. So these ideas about flexibility and dynamic range that we just briefly discussed, what if this whole thing was turned on its head? That the reason your aging brain doesn't work the same as it did yesterday might be because of the inverse, that those dynamics are shrinking rather than expanding. So the first studies we had just took a very basic view of this. We computed using fMRI data, functional magnetic resonance imaging data in the brain. We gave young and older adults different tasks to do in the scanner, from perception to working memory. And long story short, across that, the younger you are, the faster you are, the more stable your performance, the more variable your brain is overall across moments. That means the signal is expressed in a wider dynamic range. So younger, faster, better. What do we gain from this view? Right? The standard view in fMRI is really about signal averaging. So whatever those signals are that come out across trials, for example, you would just average across those two. And you can see if you compare what a variance map looks like and one of the mean bold, then you get a lot more differences than similarities. So it's not only the effect that we mentioned, but the fact that spatially what it represents in the brain is something quite complementary to signal averaging, quite different. And if you just take a straight horse race, a prediction, let's predict age from the average of those signals and the variance of them. This blue piece of the pie is what you were missing all along by ignoring the variance itself. So quite some unique variance coming through this different approach. So that's just sort of a brief taste of kind of how I got started on these topics. But the plan today is to bring you towards our more recent and future work in 3 sections. One about how this variability might represent our ability to respond to the world, how it might allow us to learn, and what the neurochemical mechanisms might be of that. Okay. So I'm a big sports guy, which as I mentioned, so all my, over all these years, all my metaphors and examples haven't had anything to do with general cognition. They seem to have more to do with sports, but that's just me. So I'm a big Steve Nash fan. If I want to talk about adaptability in the world, given uncertainty, I love this example, a career, the number 2 all-time free throw shooter, greater than 90%. So what does he do when he's doing a free throw versus a three-pointer? When he's doing a free throw, he's almost like a robot, bouncing 3 times, licks his fingers, bounce, 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 pop. He's like a robot. He says that as much on camera. But he's my height, right? So how does he survive in the NBA when he's got a Tim Duncan 7-footer arm in his face? If he goes down the court thinking, I just got to get to the free throw line, then I'll bounce it 3 times and lick my fingers, he won't score a single three-pointer, right? Ever, any shot. So he has to be completely adaptable to all the variables that come at him. So it's a completely different solution, but he then has to fit. He has to be at different angles, different speeds, different heights. Everything is on the table for him. So in the world of variance, if the demands are changing versus being fixed, we might expect that the variance of your brain as it's expressed will rise with it. And that's less so for poor performers. So our first stab at that idea was just simply comparing, staring at a fixation cross on the screen versus doing 3 different types of tasks. And in blue, you have young and high performers, and you can see that they elevate, that they start from a similar point, but elevate dramatically when those external demands are given to them, right? So when you're given these tasks, each trial is different. It's at different pace, at different elements to the stimulus, and so forth. So quite a broad thing across the brain, that the thing just opens up. And in older, lower performers, that's less so. So the next step in this thinking was basically about adaptability in general. And we focused a lot on uncertainty in that regard. So knowledge about the relevance of what you are to do might be part of this equation. But if you don't know what's coming at you, if you're a AI-based car trying to decode things in your world, how will you cue into the things that are important until you are aware of what's important? Right? What should the brain do in that situation? How should we modulate? And an interesting property maybe of the field of fMRI has been a very corticocentric focus. That means the way we think about these problems in general has to do with the cortex, and we ignore the entire subcortex. But the subcortex is pretty interesting on many levels. So flexibility studies have basically focused on the prefrontal cortex alone, what we argued in a recent review for the role of the thalamus in this equation. Thalamus is pretty interesting because it's connected to everything. It's basically a nexus for many things, but in this case, for the contextual modulation of cortical circuits directly. So everything from the thalamus flows out to cortex and back. It modulates everything from rhythms to aperiodic fluctuations. Any kind of dynamics are thought to be rooted there. But the evidence for that in humans was basically completely absent until we got at it a little bit. So if the thalamus could be a primary determinant of dynamics in general, we got to test that. And the world that we did that within is this role of uncertainty, right? What will happen to this expansion and contraction as the world changes? So the idea is that when you have certainty, when you know the solution that you have to execute, you can think of it like an attractor, right? Your system will settle on this single state. But if you have high uncertainty, you don't know what that state should be. So the idea is that you will flatten this landscape a little bit and allow a more easier transition between different states that might be optimal. Okay. So one expression of that in the brain is the so-called excitability of the system. If you have high inhibition, so your excitability is low, you typically have these high rhythmic sort of signatures. When excitability goes high, so your glutamate rises, GABA reduces, you've got many more higher frequency dynamics occurring there. So it's like a shift from a deterministic rhythmic mode to a more chaotic mode to allow for this uncertainty to be represented. So we had what we call the kitchen sink study, which is basically everything. So we measured the pupil, we had a session with fMRI, a session with EEG to get faster dynamics, and had computational modeling of behavior of a task that looks like this. So you're cued to 4 possible worlds, and the stimulus is always the same. You always have this kind of stimulus in some form. And the question is, which of those worlds will you be cued to? So if you're cued to only 2 of them, maybe naturally you would ignore the dimensions of color and luminance in this stimulus. So that when we give you a probe, which has to be one of those cues, life is a little easier. Right? If I give you all 4 cues, then you have to process all 4 elements of that stimulus, not knowing which of the 4 will be probed. So this is what we mean in this case by uncertainty. We modulate that from 1 to 4. Okay, so what happens is exactly as we hoped, which is that when uncertainty increases, you move from this rhythmic deterministic mode in EEG signals to a more excited regime, which is characterized by a decrease in low frequency power and an increase in entropy. Okay. The arousal of your system, in this case measured by the pupil, which is a proxy for norepinephrine in the system, also expands. So the system is getting up for this task, right? Uncertainty rises, you're like, oh damn, something's gonna happen, right? That's nicely coupled. And that what you do with, so when I say evidence accumulation, this is a computational model of behavior allowing that term from drift diffusion modeling. We say that basically when you what is the trade-off you have to make when you go from 1 possible world to 4 in terms of the cued states? You end up integrating evidence a little slower because you're trying to allocate a little bit to each world. Each of the 4 gets some love. But what you can do when there's only 1 world is you can quickly get to your decision, because you can ignore the other 3 sources of evidence. Okay. So the crucial thing here in terms of the thalamus, which can only be reached by fMRI, these are EEG and pupil-based signatures in behavior. We then have this other session with fMRI, and we do that within subject level as well, and couple these worlds in a massive multivariate model. And all within subject signatures, EEG, pupil, and behavior were dominantly reflected in the upregulation of thalamic activity at the same time. So the thalamus is the thing subcortically that rises to this occasion, showing you these sorts of joint expressions. Yeah. Really rooted there. We had a follow-up that just came out a few weeks ago, also with older adults. And based on these ideas we mentioned before, it played out there too, that older and poorer performers had a more muted response to uncertainty. So they couldn't easily ramp up with this difficulty. They had a sort of more similar level of all of these signatures compared to young adults. But nevertheless, thalamically dominated. And the crucial thing about the thalamus is it's quite a diverse structure, right? So what parts of the thalamus allow you to do this and that is quite varied. And these are labels for the different thalamic nuclei that are connected to cortex. And what we noticed is that the so-called medial dorsal nucleus was most crucial in this pie. And that's the nucleus that's classically thought to establish, sustain, or allow the switching of prefrontal representations depending on task demands. So this is the nucleus that was doing this sort of expansion and compression, not simple things like the lateral geniculate, which is purely a visual way station, right? So it's not a general property of the thalamus. It's more specific than that. That seems to represent how we might functionally represent uncertainty in a decision-making environment. Okay. Another form of so-called responsivity that we pursued in a few studies is basically about variability tuning. The idea is that, in theory, well-adapted organisms should be able to process the world in at least two extremes. Now, one they would call, Marzan and Dideo would call a low fidelity mode. Whenever you can minimize processing resources, you should do so. But it's just a property of fitness that probably is not controversial. But when life gets difficult, you should have the horsepower to upregulate, to do this. Right? So we have the Nadal case again, the difference between not unlike the Nash example of him sitting there with a ball machine, which has one trajectory, versus getting served to by Federer, changes in speed and direction, there's wind involved and so forth, right? The stimulus complexity of the world he has to deal with there requires a lot more horsepower, right? If you can't get up for it, that's not functional. So the question we had is whether this neural variability might be the thing that tunes to the level of fidelity required, especially in young and high performers. So we can't do cool Nadal serving studies when we have people embedded in the scanner, so things are much simpler for us at the stimulus level. And the thing that we use to get at this problem at the first pass is one of the most classic contrasts in cognitive neuroscience, which is the comparison of faces versus houses. That's done because those systems are really well mapped and understood. So it's a nice place to start with this idea. Yeah. So with things like faces, humans are experts. Faces are also very low dimensional. So you basically have two axes that you'll view a face, one here and one there, and you pretty much can get everything you need from a person. With a house, it's much less deterministic, right? You could have different numbers of windows, doors, a flat roof, or a peaked one. Anything is possible. So could it be that those who upregulate variability in line with this, right? So you're riding a gradient now from simple to complex, be the ones that can perform better. Okay. So it sounds doable, but turns out to be complex. What do we mean by the complexity of a stimulus? What is a way we can conceive of that problem to estimate it, to mathematically deal with it, and not just talk about the fact that faces seem simpler? So we took to some computational models of the ventral visual stream at the first pass. Here's something called HMax. And all it does is any stimulus you have, any picture you could show a subject in the scanner, you run a little filter through every pixel neighborhood in that thing. It's looking for flat lines, peaked lines, whatever, And you just count up. So you say, okay, how many flat lines do I have in this image? How many of those do I have? How many of these do I have? And so forth. Not too complicated. Then you just change, like the visual field does, you change the size of the neighborhood. You say, okay, now I'm gonna zoom out a little bit as I move up from the early visual system to the later functions of the visual system are more complex in their cell structure. So you just kind of zoom out, but do the same thing. So this model basically at different levels makes predictions about the types of what this image would do to what brain region. Yeah, you say, okay, we have a lot of action in these small sort of subtle neighborhoods that maybe V1, visual cortex area 1, will fire to that. But if it's something more complex, maybe it's later in the street. Okay. So it allows you a way of summarizing over a real image, a prediction about what the brain might do with it. So at a first pass, you get what you would hope to, that even across the different layers, C1 being the least, C2 being the most complex, that you have massive separation between these categories of stimuli, okay? Meaning that there's more content in the houses than in the faces. What did we find? So if we take that as a prediction, we had a sample of 46 older adults, and then mapped the difference between house and face in the variance. So if you have a high difference between those two, you have a high delta, if not a low delta. And the more you were able to expand from face to house, the better you performed on 20 offline cognitive measures from all different domains. And crucially, that effect was exactly in the region of the brain that the model would predict best separated those stimulus types in V1. So basic visual cortex, early visual cortex was the most sensitive to this expansion and contraction with stimulus differences. And crucially, no behavioral effects at all if you just do signal averaging. So the face house contrast is rooted in mean bold signals, just signal averaging over all the faces in the house and separating them. That had no relation to behavior at all in this case. All right. So far, this variability reflects the way we respond to the demands in our environment. And older, poor performers are muted in this response. If that's true, right? If it's really the case about how we bring the world in, then it should also have something to do with how we learn. Because we have to process the world, we have to encode that world to deal with it, to do something with it. So what about this learning thing? Okay. So this idea of complexity mirroring from face to house, right? We could do better. Yeah, those are two arbitrary categories that don't represent most things in life. What does our ability to respond to complexity of sensory input in general provide us? Does it really allow us to encode the world in a better way? So we have a very recent, just a 3-week-old paper or so, where we looked at this in single hippocampal cells. So now we've drilled down to the supposed center of memory. These patients are incredibly difficult to get. So when I say exclamation point after 42, that's 7 years of waiting. You had to wait for these patients to come in, then you're drilling down in the medial temporal lobe. It's incredibly painstaking work. So Uli Ruteshauser in California was the one in charge of the data collection, did an incredible job, his team. And the task is not just encoding, so not just this tricks of complexity of stimulus, but also the recognition element of it 30 minutes later. Okay. We move beyond the Hmax model to also include deep neural nets. In this case, VGG16 it's called, which is a similar kind of mindset, a multi-layer model that goes from small pixel neighborhoods to heavily aggregated ones, but one that's not built by biology, you could say, with biology in mind. So what do we do with this? At the single subject level, we compute what we call a mirroring or a coupling score, right, between their brain and the elements of the images that they see during the encoding phase. So we line up what is represented by those images, the trial level, with the spiking entropy response for that trial. We correlate those two things. So you get a matrix rho between entropy and these image metrics, the image content, and we run that through a singular value decomposition. And all you get at the end that we focus on is a latent representation, you could call it, between how your spiking entropy represents the elements of those images. So everyone gets a correlation, like a Spearman correlation basically. Out of that coupling, that becomes your individual metric that we bring forward. So we find that the higher that number is, the more you couple the image content to the entropy in your spiking in hippocampus, the better you remember those items 30 minutes later. So it's not just that you've reacted to the world in its complexity, but that has provided a downstream effect for memory and learning. And crucially, that the average spike rate, which is still to this day the de facto standard in the field of single neuron recordings, cannot account for those behavioral effects at all. Okay. So if mirroring and fidelity modes, as we call it, your ability to expand and contract, allow you to rapidly encode the world around you. So that example study is about seeing an image for 2 seconds and whether you remember it or not later. It's a totally different game if we're going to talk about learning in general, right? Most things we learn take more than a second, right? So going beyond a single shot approach to learning something that takes time. What is the role of variability in this game now? Does it allow you to get ready to learn? What happens as you step through a learning process to those dynamics? Is something we were after. And it turns out we can learn a lot from people like yourselves. Somehow, much of Nikolai Bernstein's work eluded me until very recently. But in many ways, he was ahead of the game for the types of thinking that are currently ongoing in neuroscience. So you may be, does everybody know about Bernstein? Okay. A giant for his time from Russia, talking about this degrees of freedom problem in motor control. He said there are multiple ways that you can perform a movement, and he was studying blacksmiths at the time, as well as Russian gymnasts, by the way, in the 30s, using equipment that gave them 200 frames per second in 1930. Unbelievable stuff. But he noticed right away, and they measured like heck, that these blacksmiths didn't have a stereotyped way of hitting the handle. They had all these different trajectories, arcs, speeds, what he called the degrees of freedom problem. How is it that we can get anything done in the presence of this crazy DOF? So he looked at it from a bunch of different angles, that there's tremendous redundancy in this system, anatomically, muscles and joints, kinematic, all the movements themselves. And on the neurophys level, there are multiple motor units per muscle. But why is that? What is it for? What does it allow us to do? He argued that this is an instrumental part of the whole enterprise, allowing your nervous system to explore the space of feasible actions before you get to an optimal solution. So if you don't have those degrees of freedom, you might just go with your favorite movement, but it might not be any good. Your body seems to want to continue to explore that space until you sort of settle on a functional repertoire that actually works. So it's a mechanism that if you open up in this way, these DOFs are the way that you get to the goal. So the brain, of course, is massively redundant as well. And could this thinking of Bernstein be the way we should think about the brain also? That we have to do this opening up to allow the learning to occur. And we tested, what can we do? An interesting study as well in biomechanics by Sterna, where they measure this guy with the smallest tennis racket in the world, but they have rackets and balls, and testing at the trial level how long it takes people to learn to do that. Yeah, simple task. Unsurprisingly, the acceleration of contact and the amplitude variance decreases over trials as you learn to do that. But what they do then is upset that enterprise, right? So you've learned, you've compressed, you've learned to do this thing, you get your rhythm, you have your periodic way of bouncing the racket for success. But what they do is introduce a little perturbation. So in a virtual environment, they modulate the coefficient of restitution. So the rebound, right? The thing gets dropped at a given speed, but how quickly it rebounds and how high is the thing they modulate in intensity of this parameter. Right? So you're in your rhythm, then you get dropped with a perturbation. And if that perturbation is higher, in whether it bounces higher than expected or bounces less than expected, you have to react. And then it takes you, these are like blocks of moments after the fact, how long it takes them to get back to this equilibrium. And they do that. They establish this learned thing, and then they react to something that's weird, and then they get back to action. So you're also, once you learn something, you're not stuck there. You're able to open back up, right? You're able to react to the environment. So you could maintain this invariant racket trajectory, but you don't when you're good. So you have to be able to expand again after any solution is reached. And this might be the way that we live. So we have a study where we try to get at some of this more dynamic expansion and contraction in the domain of learning. And are there any Bayesians in here? Okay. The idea with Bayesian computational modeling, the Bayesian world, if you're a Bayesian observer, is you have what's called a prior, a state belief about the world. The probability that this ball comes down and hits my rocket is X. That's your belief. And then, as you see more and more evidence about the world, you might change that belief. So here, in an example, if you're trying to guess how many bluebirds are there in the world, right? Your prior is usually pretty wide. That's showing that you have no clear idea of the probability of the number of bluebirds. And now I show you a second example. Now we have two bluebirds and one red one. Well, this prior starts to shrink a little bit. I'm converging with a little bit more evidence away from this openness to having an idea about the world. And the third example, I see two bluebirds. Well, I'm pretty sure there's more bluebirds than red ones in the world. So the idea with variability is that it would kind of track with that, right? You start from this open world at the decision-making level of not knowing what it is. We can estimate, and then compressing down as the world gets clearer. So let's see. So the task was pretty simple, kind of like birds, but instead we call them marbles. You just get 5 samples, 5 grids of reds and blues. Yeah, and then at the end you have to gauge with a little button press how many of each there were. That's it. That's per trial. So we have, I think, 72 trials, so 72 worlds that you can learn the true nature of that distribution. And then when the next trial comes, it's a whole new world. There's nothing to carry forward. Okay? So the idea is exactly that. If you start with a wide prior, where you are maximally uncertain about the world, you don't come in with any sort of assumptions about it. You just take the world for what it is. You collect data by being given these samples, and your so-called prior will shrink because you're getting to know the world. If you start with a narrow prior, you typically don't update it very well, right? You came in with an assumption about the world for which you had no evidence, but there's not much room to move there. Yeah. So maybe this wide to compressed orange case might be the ideal one. And indeed, that's true. The wider, so with Bayesian modeling, we estimate parameters over that. The prior with P, the wider the prior is before you are given evidence, the better you do on this task altogether. So starting by being locked down or having those assumptions in mind is not to your benefit. Yeah, you got to be open, collect the evidence in an unbiased way, you could say, and make your choice. And that's exactly what happens. pretty well linked. On the brain side, this compression occurs more during learning on average. So this is the average group effect, right? From sample 1, that's the first sample of each child, to sample 5, there's a real drop off there in large portions of the brain. But what we're more interested in is this estimation error, right? Who gets the task done? And people with the lowest error are those that compress that trajectory from S1 to S5 quicker. Yeah, so they ramp in a more severe way, you could say. What's interesting about the regions that reflect that is that here, the so-called default mode is maybe the most studied brain network on Earth these days. The way you can think about it in the most up-to-date way is internalized cognition. So when you're thinking about, when you're processing things internally, rather than just Netflix being jammed in your eyeballs, you're processing something about what you've seen and not upregulating your external abilities, right? So you're sort of negating the outside world while you internalize. This evolution from expansion to contraction happens in that internalized network. So the way we're framing it is the sort of development of a world model, right? A Bayesian tries to learn something about the world from this model. And this compression happens in the regions that are supposed to represent internalized cognition. So it could be a signature that you're really getting there. You've learned this thing. You've collapsed to the right solution in the right network that reflects now your real belief about the probability. And crucially, subsequently, we take that same pattern and relate it to prior width P, and it's unsurprisingly related as well, given their bivariate relation between estimation error and prior width. Okay. So you want a person that does this task well wants to start in the most open position, and the most open position in the brain is high variance. So this learning might be optimized when variability reflects the complexity of demand. Yeah. In the initial phase of learning, perhaps the best we can do is be maximally variable or open prior to being able to also compress towards an optimal solution. So you really want both, right? You don't want your brain to be forever in this chaos. Your brain has to have the ability to do the opposite. And that's really what it seems like to us now. Okay. All right, the last section is about neurochemistry. So the core idea of this whole world of aging and variance and compression is rooted in a lot of conjecture, I would say. We do a lot of studies, but at the mechanism level, many things could be happening that allow that to occur or not. In the world of aging and performance, dopamine is the first stop you would dig down to. You know, for many years, that's been the primary mechanistic model in that field, where, for example, in animals, you show a 10% reduction in dopamine receptor density per decade between 20 and 70. As older adults generally exhibit less signal variability overall, it's plausible that that might be due to these dopamine system inefficiencies we expect to exist. So in a mouse model, they have very interesting ways to manipulate it, of course. So you can take dopamine gene knockout mice who have no endogenous source of dopamine at all, and then give them L-dopa, this precursor to dopamine, and allow them to synthesize it, but only from that source. So you can see when you knock dopamine out, you get these very stereotyped almost single spike profiles that are missing all the dynamics you see in a normal mouse. And if you provide L-dopa to them, you gain some of that back. You get a bit of bursting, you get spacing in the timing, lots of things happen. So we took this quite literally as a model for aging in that sense, and gave young and older adults amphetamine in a low dose during the working memory task. So it's a pretty low dose, but it's definitely enough to drive that system. And on the SD bold level, on the brain variability level from fMRI data, you can see what we normally see, that young adults have more dynamics than older on every task. And if you give the drug to young adults, you don't see much movement, but they then again already have optimal sorts of amounts of dopamine expected to be there. And in older adults expected to have a low amount, we see a dramatic change there. So the ability to expand the dynamic range of their systems is quite easy, actually, even in low dose. And that happens in some pretty dopamine critical regions, in the striatum, for example, and in the prefrontal cortex. The working memory task, these would be like expected, you could say. But really, we're driving the variability in some of them by 30%. Yeah. And crucially, if you couple your movement under the drug, how much you reacted to it, you can see that older adults in particular were responsive as well, that they got faster when their brain variability rose under the drug. And they also got more reliable in their reaction times on that task when they expanded upward on the drug. We can go beyond dopamine as well. There are many neurochemicals in the brain. Maybe one of the more obvious places to stop if you're going to dig there is GABA. GABA being the most important inhibitor in the brain. It is absolutely required for moment-to-moment dynamics in models and animals. And you can see here, if you antagonize GABA, if you reduce the amount of GABA in the system, you will lose entropy in the expression of those cells here in mice. And maybe one easy way to cartoonize it is you have a lack of dynamics at both extremes, actually. So if you make the system hypo-excitable, you over-inhibit it, and you just hit the floor, basically. You've eliminated the ability for those cells to fire. And the same is true at the other end. If you reduce all the GABA and you just pump it full of glutamate, then you end up in the opposite extreme at the ceiling. So in the middle, where balance is existing between GABA and glutamate, you have the ability for dynamics to exist. Yeah. And it's well known that older, poor performing animals exhibit lower GABA overall on every level. So can we manipulate that? We started by doing that in resting states and no task involved, and that worked pretty well. We can boost signal variability at rest in older adults on a low dose GABA driver, lorazepam. That works. But what about the function of it? Yeah, that's just a proof of principle of the drug, you could say, in the variance domain. We're more interested in the human performance level of it. And GABA is crucially required for visual tuning. So here in a monkey study, a key one from 03, you have young and old monkeys. You see that young monkeys, these are sort of profiles of the specificity of a cell. Yeah. So the response profiles for visual cortex cells. And you can see that if you manipulate GABA upward, they already have a lot of it. It doesn't seem to hit them too hard. But if you reduce it a lot, then they look a lot older. And in reverse, there's not much specificity to the visual cortical cells that they measured in older monkeys that control. But if they boost GABA up, they can bring this profile into a more specific domain. So something crucial for the visual domain in any case, in the context of aging for GABA. So if younger high performers can upregulate variability to allow the encoding of this image complexity in their environment, could it also provide a window into why that is? No. So we tried to do that. You can measure GABA in the brain by magnetic resonance spectroscopy. They're pretty big regions you have to measure over, but you can do it. It measures the spectral properties of the signal that comes out of the scanner to isolate what GABA would be. And then we give those people a low dose lorazepam again. So the same drug manipulation as the first study, but now on task of viewing faces and houses. So we have these expectations about the complexity of the stimulus. We fit those models again and run it. But the key here is that here we measure GABA in the visual cortex and represent bold variability there. So we've married those two. And you can see that the more SD bold relates to house versus face, so the higher that is, the more you've upregulated, expanded, as you got to the more complex stimulus. the higher your visual GABA was as well. But the key, that's in young and old, showing that effect. But the key in all the neurochemical, sort of neuromodulatory elements of cognitive neuroscience in humans is the expectation that what the drug will do will be a function of what level you were at. But usually you don't measure what the baseline level is before you give them a drug. You just give these people a drug and things happen, some go up, some go down. You say, well, that's because this person was low, so that's why they went high. And this person was high, and that's why they went low. Yeah, so the classic inverted U problem, but there's been absolutely zero human evidence for that. So in this case, we had both. We had MRS to measure baseline GABA, and we had the low dose drug. And we see exactly evidence for that inverted U. And if you can spot that, so someone who starts low would go high. And this is the GABA drug related shift. So if you go high, right, the people that went highest started with GABA the lowest, and vice versa. So even though it's a linear trend, that's the expression of the U in regression terms. Everybody, is that clear? Okay. So the people down here have gone below zero, that's dropping. People on this side have gone above zero, that's rising on the U. Yeah. So the first evidence that we're aware of really anchoring these drug effects by the level that you start at. So last thing is about the future of this part of our program. In a recent review that just came out, we sort of lay out what we call a functional future, because this cognitive neuroscience of human aging is typically rooted in the structure of the brain, by and large, and not about how older adults do and don't perform well, right? And the brain areas that represent that. So part of that functional future that we've laid out, your vision, is about combining these different approaches. Usually if you're a dopamine person, that's what you study. If you're a GABA person, that's what you study. How can we bring the worlds together? And already a quarter century ago, Segovia and colleagues, they talked about this in rats as being well known. You know, that there's no understanding the dopamine element of this without pairing it with GABA. And in fact, it's the relationship between dopamine and GABA that gives you all the apparent dopamine effects that you can find in the dopaminergic parts of the brain. So we're trying to marry these worlds, and at the design level, that's not trivial, but we can manipulate GABA with these designs that we have, and could collect dopamine PET, for example, pre and post drug. We could try to predict the GABA response from pre and post dopamine data, and vice versa. Baseline GABA could be a predictor of the ability for the system to respond to dopamine drugs and get us a little bit closer to these designs. Can be complex, but they are doable now, they say. So we have to kind of abandon this tendency to silo ourselves, thinking that we have to understand only one thing at a time. It'll probably never get us to where we want in the domain of neurochemistry, at least. So in the end, what the group is trying to do is understand the brain in its natural, ubiquitously variable state. How to measure it, how to manipulate it, how it's built, and when it's good. And with this 15 years, we've tried to sort of promote the idea that it's not this noise that you have to deal with, but that it provides a signal of its own type, of how we can optimize human performance, how we develop and age and beyond. You know, really taking it seriously. Thank you to everyone, our many funding sources and the NPI in Berlin, and to you all for the chance to be here today. It was really great.
That was a great presentation. Thank you very much. I saw something on one of the slides where you had SD bold, which maybe if you wouldn't mind as part of your answer, just explaining exactly.
Yeah.
Try to figure exactly on how you measure that. But there was one graph where you showed older adults and younger adults. It was like the older adults were clipped at 0. and then the younger adults seem to have like a range that like far extended out. There's one of the graphs where you were showing like the two of them on a plot, SD bold, I think was on the horizontal,
Was it the GABA?
a little earlier.
Okay.
Uh,
But it was young and old?
the two regression lines on it.
This? I'm not sure which one you have in mind.
Well, yeah, anyway, maybe I'll just ask the question differently, is like, I noticed that it seemed like based on what I saw that the SD bolds, this measure of variability, seemed distinctly different in older adults and younger healthy adults, almost as though like, I mean, if I interpreted it right, like, yeah, older adults were really clipped in a really narrow range. And I guess maybe if you could just comment more on what drives that and exactly what SD bold is and what it means.
The individual differences you're commenting on, the compression of the group,
Yeah,
is that what you mean?
Yeah.
Yes, there are some experimental scenarios for us so far. It depends how well I think I know what you mean now. So, the brain score, these multivariate models are a little bit complicated to describe in short order, but you have a series of weights on two sides of the equation. In this case, let's just forget the rest of the variables, EEG and fMRI. We call them X and Y. And the weights are generated from both sides to maximally express the relationship between the two sides. Now, if the older adults, so the scale here is at 10 to the 4, so it's a little bit, just for pictorial purposes in the paper, because we didn't have enough room to expand to the full value range. But there are values there, so it's not really zero. They're in the thousands there actually. But it's the integration, the dot product of the weights and the data. So there is no zero per se, because the variance can only be positive. There's no way to have negative variance in this case as well. So I think, yeah, but I can show you if you're interested in the model afterwards, I can show you the details more if you like. Yeah.
Yeah, so I recall learning a long time ago that the eye is always fluctuating, right?
Yeah.
And that the
It's a microsuggest.
yeah, micro movements, and if it ever stops, you actually lose your ability to see something,
Mhm.
right?
Mhm.
And it's actually that movement that gives us the ability to see. Do you see this loss of noise in the brain as it ages reflected in the pattern of the eye and its movement?
Yeah, we haven't published that, but we have a study with complexity of stimulus with eye tracking with fMRI. And that's exactly what it is. So they end up, it's kind of a unique thing that they do. They go to more positions in an image. An older adult will go around the image a little bit more, but in a more like random way that doesn't seem to help them. And if they're more targeted means that when they land in a position in the image, when they're going to perceive what's there, the young adults will actually stay for a half second longer. You know, they'll just go to that position. Okay, got that. Yeah, got that. Then they zoom out and can see it all together as a unit. That seems to be the behavior. So they do move around, but almost too much. But this is the pupil now. Microsaccades would be more about the scan paths. About them and the dynamics there are a little bit different. But yes, they do something suboptimal with their eyes as well. And so you can force, there's some designs where you can force old and young to look at an image in the same way. You just put goggles on them and then you give them like this zoom view and equate the scan paths of young and old to see the changes memory. It does have an effect. But why they don't do it naturally is not really well known, I would say. Yeah.
Adam, Reza, and then Venus.
I had a question about some of your ways to define the image complexity. So I noticed you use things like filters and max pooling. And I've only seen those in sort of neural network implementations, so it's cool to see how you managed to use those in this situation. But I'm curious how things like spatial pooling and max pooling are represented in how we view images. Like I think I might have just missed sort of the translation and
Yeah.
why this is applicable here.
So this is truly like a visually inspired model, whereas the VGG thing, any modern deep neural net that is used in machine learning of images or object recognition, I think it has no brains in it at all. They go around the other way and bring it back to the brain in terms of a brain score. That's a long story. But this was really rooted in the 90s already with the visual cortex in mind. But you can see why the other models also work, because the visual cortex basically gets, you could call it less specific as you move up the visual stream. Yeah, so you have, if you go from V1 to V4, you have all these stops in between. And the reason they frame it this way, the pooling is a mathematical way to represent probably what the brain does, because the cell sensitivity, if you measure like V4 downstream, which is supposed to be more specific for complex cells or more abstractions, you could say, something like this, versus the pixels themselves. And you give it a pixel, you measure in V4, but you give it a pixel, you won't get much of a response. So the response profiles from V1 is basically looking at what you could call the pixel level of life, right? These very fine detailed elements, which of course have to be aggregated, summed over, integrated, however you think about, to get the whole shape of an object. Like imagine looking at me right now, right? You can't imagine me just with these 45-degree lines. You have to integrate all this stuff eventually to make the whole. And that's what the whole system is built on, that it's passing what you could call the pixel level along the chain. Okay, so here we detected all these pixels. Okay, then I pool over them, or I max or sum over them. Then I get these kinds of outlines eventually, which is made up of thousands of those arcing elements, right? It has to be combined. So that was their motivation, I guess. But the reason the DNNs work is because it's basically the same kind of property, right? In some other mathematical way. You just say, I'm going to take these fine-grained image differences and I sum and integrate and pool and do all these things depending on the model. And at the end, I end up with an ability for that model to tell me that that's a cat, right? But it's made up of everything. Yeah.
Reza.
Very interesting presentation. I was wondering, you showed medal that was waiting for the ball and was moving.
Yeah.
And you showed that it shows variability. I was wondering when he wants to hit the ball, that variability reduces or still
Yes,
much variable.
definitely. There's one other study that I wanted to get in the presentation today. That is about baseball. And that's exactly what they do, is train people to hit a ball through highly varied training conditions and compare that to people that haven't seen those varied conditions. And these are like the phases of the swing. And they differentiate between landing and weighting. Landing is like when your foot first hits the ground, they measure that moment. And then the weight that's applied is after so many milliseconds, like what is the force? So they're on a force plate. And you can see that as they continue to be driven with the variability of conditions, that's different speeds, change ups. I don't know if you know baseball, but all the different pitches you could receive are shown to you while you train. And two crazy things happen. One is that the variance of, so now you're post training, right, in white. The landing and waiting phase goes up in variance. That means you have more in your repertoire of when you're going to plant before this comes, right? But when you're in action at the swing and what they call bat and hitting zone level, it is quenched. So you have come up with the thing, right? Because you're reacting to this, the difficulty of whatever that pitch is. And the best hitters just adapt like heck, right? All the best hitters in the history of baseball do that extraordinarily well. It doesn't matter how fast, left-handed, right-handed, they don't care. Why don't they care? right? It's because they're doing something like this, maybe.
And is the intensity of the variability different from one person? Because there is variability, but also intensity of the signal.
Definitely. Yes.
What does that indicate?
So, yeah, the difference, the way we started with these mechanisms, like the neurochemical things, you could estimate, like you might have twice the dopamine that I do. And you just live that life and I live mine. So when I'm given all these, all this uncertainty in life or adaptability that has to be forced on me, I may just not be able to react like you can. So the base rate of variance does have, at least computational modeling would suggest its elements. Right? You have a certain amount of GABA, a certain amount of dopamine, a certain amount of glutamate, which gives you your base rate. And maybe that allows you to react better than me because I don't have those same levels. So there is definitely relationships between baseline brain things and these neurochemicals, for example. You could think about it at the latent level of performance, just someone that's good. We see over and over, like I mean, dozens of different tasks. That your performance, if you're just a high performer, regardless of the brain measurement having anything to do with those things, right? I just put a battery of tasks to you, you do 50, 60 of them, and then I relate those back to what your brain does at rest. Those relationships are there.
Just one last question. You showed a figure SD, VS cognitive performance. And there was such variability in the data point with the aging. I was wondering if that variability is due to the method that you are measuring those points, or it was just some older adults perform the same as younger adults.
I'm not sure which one.
This delta SD VS cognitive cognition.
The GABA stuff.
No, it's the first project that you have.
Okay.
Delta SD VS, no, just before. It's a line that you feed to the point, so it's very early. Delta SD and cognition. One of
I
them.
mean,
Yeah.
this is quite late, but maybe that's the one. Okay.
Yeah, exactly.
Yeah.
So some of the older adults just
These are all old.
all
In
over.
this case, yeah, it's, well, 60 plus.
Okay.
Yeah. That's right. So we summarized over 20 offline cognitive measures to make this latent cognitive performance score, but the same holds for each of them.
Is there any uncertainty in the method that you measure these points, or is just, this is all delta SD, absolute delta SD?
I'm not sure what you mean, but basically we have a pipeline where we get the variance for each condition. And that has its own sort of baselines done to it. And there is a series of steps that go into the estimation of the SD, but once we have those quantities, we just subtract them. And then they go in the multivariate model to find a weighting pattern spatially that represents cognition. But yeah. Well,
Okay, we have Venus and then Aaron.
Hi, Pip.
Hi.
Actually, you touched a little bit on it when you answered the question about the neurotransmitters. It's like you're saying when we get older, there is damping of this good noise and all of that. So how to prevent that? That's one question. And usually dopamine, you say exercise, like it's good to have high level of dopamine. So what are your thoughts on that?
So on the drug side, I mean, nothing works. You can, yeah, of course, we can give them amphetamine, but the runway for that drug is low, right? So short. Your body will acclimate real quick. Yeah. So,
Naturally, no drugs.
yeah. So all the drug studies so far have been about pointed sort of proofs of principle. We haven't done much ourselves with how to drive dopamine with fitness, for example. That's not so common in our field, at least. There's a lot of fitness studies, but what they do with the brain is sort of like an afterthought, in my opinion. Does the brain avoid shrinking? Does the brain look differently in some network way at rest? This kind of thing. But there's no real targeted sort of human performance element of this, right? What is that element? And typically that's the case we made with this review at the end that I mentioned is It really has to be about how the brain does that performance. You can make all the claims you want about the trait level aspect of it. Someone who is this good on task A also has a brain that looks like that, but we really want to marry those worlds. So as far as I know, there is nothing. Could you do a clinical trial, for example, if there's a manipulation that is cognitively relevant, you know, in a way that you could make predictions about this much, much harder.
But for example, the subjects that you had, like the older adults, did you ask them about their lifestyle?
Yes, sure.
They exercise, that they were all sedentary people, or they were exercising during their life, or like something like that?
So in these studies, not. Other studies at the MPI, they have done that sort of in a more targeted way, even with fitness regimens that they go through while they, in a longitudinal way, track them over 2 years, for example. We haven't done that in this domain. So, but I'm not aware of anyone that has evidence that, for example, dopamine can be driven up by this. You might find the correlation. So typically high performers in our hands, they're a little bit better educated. They are a little healthier, but all the people we get into the lab are typically pretty good. You know, they're all retired, they're all healthy. That's a different game, how to properly represent older adults in sampling. But I would say nobody does that so well, except at the population level. But yeah, good point.
Okay, Garrett has a question?
I was just curious if you've looked at the variability throughout the maturation process.
With kids, you know.
Yeah, up until from, I guess, 0 to 20, looked at 20 and older.
Yes, we have started to. You want to just say a little bit about what we see here?
Yeah.
Okay, yeah, it's somewhat different, depends on the type of variance you look at. But just take EEG, for example, where the waveforms can be much better expressed than in fMRI. So, what happens as you go from childhood to older adulthood is when you have that, I'll show you what I mean. Okay, when you do this spectral thing, right? So you just take any signal and you represent it in terms of the Fourier or wavelets, whatever you have. You have low frequency stuff, slow waves, and fast waves. And if you express that as a slope, you have something like this gray line. Okay? So we call that the spectral slope. And what happens from kids to adulthood is the increasing flattening of that thing. So kids have a lot of low frequency content and not much high frequency content. High frequency content is expected to represent your perceptual abilities a little bit more, for example, than the low frequency. So you just, if you tilt that a bit to sort of young adulthood, then you get a better balance of this. And in older adulthood, you lose this low frequency element even more and end up in this high frequency domain, which means that overall, so this is in log, right? When you do these spectral things in log, the low frequencies are dramatically more represented than the high frequency stuff, even though it's a line. It's sort of an artificial line because you log transform. So the low frequencies then are dominant in children and absolutely absent in older adults. Yeah, so it's a totally different construction of the variance, you could say, across all of those things, tilting towards high frequency.
We had one question here and then we'll take a last.
Hi, the noise pattern that you found between the young and the old people, is this a difference as well as the different activities in the young itself? And
I didn't catch in the what?
in the young people, so when you have a noise pattern, right? The noise, the brain noise, you said. Yeah, and the old people, you both see that in the old people, the noise pattern is slightly higher.
Do you mean with this?
The noise I'm talking about. The noise.
I don't know, what do you mean when you say the noise?
The brain noise, you said, right, first. The brain noise in the young and the old adults, you said that the old adults have a higher noise pattern.
Typically, it's the opposite.
The opposite.
Yeah.
Okay, so the question,
Depends how you define it, but
yeah,
overall variation shuts down. That's what you can say.
okay.
Yeah.
And does this differ from activity to activity? Let's say if we do only with the young people, does this differ from activity to activity, the patterns changes or? it remains the same?
Yes, it seems like even in older adults that depending on the design, so I'm gonna show you a few today, but you really can get them to move as well. So the demand is crucial, it seems, to everyone. There's almost nobody that flatlines really in a numerical way. They will just boost a little less or a little more kind of thing. And in some cases, that's extreme, depending on how the demand might be. So if you make the task really hard, what we talk about this cliff idea a lot, right? You might be very good and you can elevate or expand your variance in line with demand. Demand is increasing, so you're rising to that occasion. And then over here, you might hit your own personal limit and just fall off. But for me, I might do that here. So I might have no ability relative to you to do that, but we all probably have that ability, right? So far in the data, numerically, it seems that way. It's just whether it's statistically boosting or not in everyone.
So I thought we had one last question, but we have three, so
Yeah.
we'll do all of them, so right here first.
So just confirming, neural variability decreases as you age, right?
Yes, in an overall sense, yeah, we can be nerdier than that about the measures, and but you could say that.
What do you think, like energy availability within the brain, do you think that plays a role? Because when you age, usually that decreases as well, right? So glucose transport slows down and stuff like that
Yeah.
slows down.
That's great.
So then what do you think energy availability plays a role within the neural variability, or how variable you can be when you judge certain tasks?
Definitely. So we unfortunately didn't get too far on that level because it's really hard to do energy metabolism in the brain, like to measure it in real time at a spatial level in a given subject. That's pretty tough. But we started a PhD, one student doing exactly that. We had to design all the scanning protocols, the actual sequence in the MRI itself by hand, and we just couldn't get there. But the preliminary data looked very good. So if you measure creatine, for example, you can do it. It's really spatially crude. You almost get like the brain cut into 4 pieces or something. But you can, with your eyeballs, see in a given subject, if you do at least 2 minutes of scanning, that visual stimulus, you just give them a visual grading, something that really, like a strobe almost, that drives the visual cortex crazy. That's the easiest way to do this kind of thing. If you give that stimulus, creatine goes up, and then if you give them a fixation cross, so nothing happening, that shuts down. It takes that long to even measure 1 point, but in theory, it can be done. And theory-wise, in expectation of that, you're absolutely correct. So the ability to the energy demands of cognitive demand are probably exquisitely clear if we can measure them. We just can't.
Three and then Bella.
Great presentation. I was just wondering if you've done any work looking at sex differences. And if not, what we might like speculate would be differences.
Unfortunately, not really. We've looked in a few data sets whether there were any differences among people that we had sampled, but we didn't do it in a targeted way. We just took males and females that we had. In some cases, we had controlled for that variable before publishing it, for example. And none of our primary effects changed, but we have colleagues that are doing it properly scientifically in McGill, now in Toronto as well, with variability in mind. So, but no results there as far as I'm aware. I think they have a lot of ideas that it should be different than at different stages of life, because the group, sometimes Natasha Raja is her name, she's at Toronto Metropolitan now after being at McGill, has clear aging related ideas around that as well. What's happening through post-menopause to the ability to up ramp to demand and stuff. She's studying memory in that regard. So not much to say. So yeah, good point.
Okay, the last one from Bella.
You talked about learning. And we got some people learn optically, some acoustically, some slow, some fast. What is the difference?
Oof. Like on the individual difference level, what makes me need both vision and sound when I learn something. And for example,
Yeah, some people, they, I mean, you want to go from A to B.
Yeah.
And some people go once and then they know it. And other people never keep it.
right?
But
Yeah.
it's the history.
That's, I mean, probably a million answers to that question. I think if I just focus on the way you framed it at the beginning on the sensory level, there are people that prefer those things. The people, we've run one, complex study on multisensory integration. That there seems to be both in behavior and brain, this very present boost when you can have two sources of information. Even when they're not perfectly aligned, it can boost you. So let's say you had to choose right versus left. And visually, when a square goes on the right side of the screen, you're gonna press the right button. And when you hear something in the right headphone, you're gonna press the right button. Then we modulate when those two sources of information come into the system, right? We can delay them. We can say, okay, you might be a visual person. So if I give you the visual one first, and then I gap the auditory one enough, then you might not even need this one anymore, and vice versa. Yeah. I don't think people know necessarily what their optimum is, but there's an awful lot of people that despite your brain having to deal with two sources, they gain a lot from that. So me as a learner in undergrad, I always had to, I had to hear it for some reason. But I'm a music guy. I've played music my whole life. I don't know, maybe that's it. But the reasons for those individual differences are really poorly understood. The presence of them is clear. So we had distributions of people that really were visual or auditory people, and you could boost them a little bit by adding the other thing, but it wasn't nearly like a person who was kind of tuned to integrate multiple sources. So the guess from that study is that if you can integrate multiple sources and get something out of this, you might be the best learner that exists. However, it might be a crutch at the same time. If you can't do it without all the sources, then you have to create those sources. And that's harder to do, I guess, in real life. Yeah. But in terms of how the brain does that, that's a career, I think. Yeah. Good point.
All right, with that, let's thank Dr. Garrett.